Uh, Jordan was my PhD student between 2004 and 2007. And during this period, he determined the structure of the extracellular domain of the receptor for interferon, which was a very challenging project for that time, considering the 25 kilodalton size of the protein. He also studied the interaction between the receptor and interferon itself, which form a 43 kilodalton complex. Jordan was not only an outstanding student, but he was also a great colleague for me and to all other students in the lab. And why is that? Jordan is very knowledgeable in NMR and not only in NMR, and was really superb in helping other students when they encountered problems understanding some principles or experiments. What is special about Jordan that he did this really passionately, and he did not intimidate the students who asked questions that were sometimes stupid, and he had extreme patience, too much patience to my taste, and great willingness, and his explanations were very clear. I believe that I don't want to offend other people, but in this respect, he doesn't have any competition at all. No wonder that he is such an excellent lecturer at Bar-Ilan University and well known for the qualities of his lectures. He then did his postdoc with Adbax, where he studied the KSCSCA potassium channel, and in 2007, unfortunately for me, he moved to Bar-Ilan University, where he became an associate professor in 2014. Jordan research focuses on membrane proteins and intrinsically disordered protein. His lectures in scientific meetings are always excellent and very well accepted, very clear, and I'm looking forward to his talk today. I should mention also that Jordan is an excellent piano player and maybe he competes with Christian in this respect, but he has also something else that Christian cannot compete with him, and he is also a cantor in his local synagogue. So I don't know if Christian is a cantor in the church or something, but okay. One problem with Jordan, I understand every word that he speaks in Hebrew, but sometimes I have problem understanding some words in English, and maybe he should explain it to us also the title. So the title of his talk is Evasive Dropping on the Cellular Advantages of a Pathogenic Cytotoxin. And actually I have to go to Google in order to understand what is evasive dropping. So I think your task is to explain to us what is also the meaning of evasive dropping. So please, Jordan.
Well, thank you. Thank you, Yaakov, for that kind introduction. You certainly set a very high standard that I'll try to meet in the coming talk. And the title actually does begin with the word eavesdropping. Eavesdropping, which means, for those who don't know, but probably many do know, sort of spying on or trying to hear something that you're not supposed to be hearing. And it's a reference to the fact that a lot of what we do in high resolution NMR today, as you know, as all of you know, is often to try to be a silent observer and see the things that are happening in solution to proteins that other methods aren't able to see. And NMR, just for those who are from other magnetic resonance fields or even other fields entirely, the solution type of NMR that I and many others run is sort of a combination of the biophysical methods in the sense that it looks at the protein in in a very realistic, in a very natural environment, but also it maintains that molecular local type of data that NMR can give us, where it can give us information on an atomic level basis, essentially. So when I explain to colleagues, and I'm often in this position of telling what NMR is about, it's like those other methods, like fluorescence, like many other methods where you have a probe, except here we have a probe almost on every atom if we want, and that's what gives our method a lot of its strength, if you like. So we're eavesdropping on a process that we have no business being there, but we will try to learn something about how a cytotoxin, an effector called BTA, which comes from the pathogen Bordetella, learn a little bit about what happens with it in the cell. And we're definitely only scratching the surface here. I will now share the presentation. We're only scratching the surface, but I'm hoping that even what we're showing, what we're giving here, will be helpful, not only to learn something about the pathogen itself, but also to demonstrate, I hope, the power of NMR in studies of this type. So now I will share the screen. Okay, I hope you can see the screen now. So the topic is, as I said, we're eavesdropping on the cellular adventures of a pathogenic cytotoxin. It comes from the bacteria Bordetella, which causes pertussis. And we're going to be focusing on what happens in this system here. Let me just get a pointer going. This system here, which is known as the type 3 secretion system, which is common to many pathogens. And that's why we're hoping that what we can find in this context is actually not only specific to Bordetella, but also might have important impact and ramifications for other studies. And as you can see here, the system is made of 3 parts. It has this needle, which, as many of you know, there was some very nice solid state NMR work showing the structure of this needle. But through the needle, we need to inject an effector, which is shown here, this part right here. And there's another component to the system, which we know as a chaperone. The chaperone that binds the effector while it's in the pathogen, and it sort of primes it for injection into the host. And in the host, which is what we see here, is where the effector starts causing damage. Before I go further into the presentation, I just want to make sure that I remember to thank and acknowledge the people who contribute to this work. So this work is really the PhD thesis of Adia Halom, a very, very talented PhD student who's graduated about a year ago. She continues to do well in her present endeavors. So this is basically a big chunk of work which is performed by her, including some of the initiation of the experiments and the ideas of the things we wanted to study. Dr. Hadassah Shaked is our always reliable and always involved lab manager, and she was instrumental in making this project work. I really have to mention a brave, actually undergrad student, Miriam Drory, who just a few months ago expressed an interest in doing something in the lab, and I said, why don't we do relaxation dispersion calculations, which apparently didn't daunt her, because as you'll see, we got some very nice results. So thank you, Miriam. I'd like to mention the Ben Gurion University group of Raz Zaribat, Professor Raz Zaribat, who actually came to us with this protein about 8 years ago, promised it couldn't be crystallized, later reneged on that promise, but more about that later. And of course, their collaborators, Geula, Sophie, and others. And also, we benefit tremendously from the collaboration with Professor Sharon Rothstein from the EPR group here at Bar-Ilan, and the NMR facility manager and assistants and helpers. And as you know, there's a lot of work that goes into making sure everything runs smoothly. So with acknowledgements out of the way, let me get back to our T3SS system, which you see here. And now more in detail. So we have 3 types of components of the system. We have the structural proteins, which form the needle. We have the effectors, which are the ones that need to be injected into the cell. And we have the chaperone, which is known to bind to the effector inside the pathogen. Bordetella itself is known to have 5 different effectors and 2 chaperones. This is what's known. And it's also known that BTA, shown here in blue, and BTCA are what we call a cognate effector chaperone pair, because they're known to bind together. BTCA is the chaperone of BTA. Previous studies of BTA, which actually commenced when Raz came for the first time in touch with this protein, what you would call medium resolution structural studies, not a structure yet, but just characterization of the system. And we had a certain part in this. Here you see a Sachs characterization. This is of a 287 amino acid fragment of the protein, which overall is about 650 amino acids. So this is cutting off a piece. And also, we performed an NMR study of how the chaperone binds the effector in this form of the 287 fragment and what it does in that situation. And we were able to see certain effects, but admittedly, the system was quite large for NMR standards. And this was, as I say, the protein that Raz claimed would not be crystallized. So let's see if NMR can say anything about this. This is the point we're at now is maybe around 5 years ago. So we set out to analyze by NMR a smaller fragment of the protein, which is known from structural and from also various studies, was shown to be the business end, if you want, of the effector. It's an 131 residue N-terminal fragment, and it's about obviously much more amenable to NMR studies. It's smaller. It is a helical protein, so we weren't expecting great dispersion in the NMR spectrum, but you can see you get relatively good quality spectra here. This is measured at 700 megahertz at about a 0.3, 0.4 millimolar sample, 30 degrees, or you can get a good spectrum at 10 degrees as well, which we often do. And what you can see here is the very usual analysis showing helical regions, loop regions, everything is behaving as expected until one day Raz called and said that we succeeded in crystallizing this fragment. This is the smaller fragment which they were able to crystallize, which luckily, since we're working together, rather than scooping us, essentially propelled us forward to give a more structural context to the findings that we were obtaining using NMR. So just to show you what we're looking at, it's a 4 helix bundle. You can see the 4 parallel helices shown here in gold and yellow. There are 2 other smaller helices. We call this helix here the D helix. We call it the tip helix. And in general, it's a very elongated molecule, ratio of about, I would say, 4 to 1 of its 2 axes. Here you can see in this more detailed structural picture, you can see the hydrophobic core. So the helices are pointing the hydrophobic residues into this bundle, as is typical of these types of proteins. Another important feature is the fact that when you look at the surface of this protein, you can see there's quite a bit of charge separation. So the protein isn't particularly charged in any direction if you look at it as a whole, but it does have a more positive side here and a more negative side in the back, which is, you can see it peeking out here. Now, going back for a second, just to place these things in context, looking at this diagram of the overall process of an effector being injected into the cell, 2 particular questions stood out and gave us a feeling that we had a chance to perhaps address them using our NMR methods. One of them was, and I'll go in reverse order chronologically, one of them was, what does the effector actually do in the cell? And it's known that the effectors basically target the host membrane. Once they bind the host membrane, they kind of hijack the mechanism that makes the membrane work, or makes the cell work, and use that to their benefit. And that's the beginning of the infection, if you want. So one question we asked was, how does the effector get to the membrane, recognize the membrane, bind to the membrane? And the other one, which comes first, but in the talk will come second, was what is the nature of the interaction between the chaperone and the effector? Since we know the chaperone is meant to guide the effector, if you want, to the needle complex, but also while it's in the pathogen to perhaps keep it in a certain structure or stop it from interacting with other pathogen proteins. So that was the other question we wanted to address. And as you might have noticed that this project doesn't really fall under either of the 2 main topics that we pursue in the lab, membrane proteins and IDPs. But I found that as we go into these projects, often what comes up is the ability of NMR to Look at the plasticity of proteins if you want, and relate these changes that proteins undergo eventually to their function. So I think you all agree that what we call the structure function dogma is really, there's something in between. It's not only structure function, we know there could be dynamics in function, or various structures in function, or conformational changes. These things have become very common to pursue, and NMR is very well positioned to be looking at these types of questions. So therefore, even though this is not a membrane protein, but it's a protein binding to a membrane. So we routinely make in our lab membrane-like assemblies, preparing what we call a membrane scaffold protein, wrapping it around lipids, and embedding a membrane protein into it. Actually, if Andy Bird is listening, I couldn't catch if his name was there on the list, but this is an idea that I got from him because he presented at a conference, I believe it was in Kyoto 4 years ago. He was describing how you can really look at binding to membranes using these membrane mimics if you want. So rather than embed a protein in the membrane, we're going to prepare a little piece of membrane and see if the protein binds to it. And that's what I'm showing here. So if you take the spectrum of our effector and you add a nanodisk, which has only, in this case, BMPC lipids, so it's just the standard lipid, doesn't have anything special in it. You see that the addition of the nanodisk doesn't create any change in the spectrum. But if you dope that piece of membrane with PIP2, which is actually the part of the membrane that the effector is known to look for, a negatively charged lipid, then you can see changes in the spectrum that result from the addition of this membrane mimic, which you can think of, if you want, of a membrane where a hole was punched out of it, and instead of using the hole, we're using the little punched part, and then wrapping a little rubber band around it that keeps it from touching the hydrophilic surroundings. And that's really what we have here. And if you look more carefully at these changes, you can see that they're localized into certain areas of the protein, and that can be mapped onto the structure. And that immediately showed us that indeed this positive region is in charge, is responsible for the binding to the membrane. In order to pursue this further, we realized that it might be easier to just look at the actual PIP2, because making these membranes is quite tedious and takes a long time, and you need high concentrations in order to perform titrations like this. So it occurred to us that maybe we could get away, if you want, with a slightly different approach, and that is, let's just use a soluble version, a soluble analog of the PIP2, which worked actually quite well. We convinced ourselves that that's the case by comparing the results, which I don't show here, between the soluble PIP2 and the membrane. And once we saw that they were quite identical, we continued on with the soluble one. So what I'm showing here is, let's find out which residues exactly are in charge of membrane targeting, are the ones that bind to the membrane. And you can see here, in the case of wild type, the changes that occurred as a result of adding the membrane mimic, this thing, to the sample. And then when we mutate various positive residues, here, and here, we chose a few of them, you can see that they create different effects. Of course, we wanted to make sure that none of these effects were actually equivalent to the protein unfolding or falling apart. So for that reason, we have here in parallel also the spectral changes that you get when you perform these changes. For example, it's easy to see that when we change our arginine 100 to a negatively charged residue, then we really can't trust our results. It looks like the protein is undergoing some global change, so we don't look at this. But when we see a case like this, where it really looks like the change is very, very local, only around the mutation area, we feel that the fact that here we have binding and here we have a different pattern is actually quite significant. So as a result, we were able to pinpoint that actually residues R59 and K62, these two positively charged residues, were the ones in charge of binding to the membrane. Remember, they're binding to this doped membrane that has a bit of PIP2 in it. And you shouldn't think that this is trivial, because actually other studies showed that toxins, similar toxins, homologous in structure and from the same family, actually tend to bind to their tip. That will be this region here. So here's an example taken from a paper on Clostridium difficile toxin. And you can see that this toxin actually chose to bind through the tip, the region here, which in our case just doesn't happen. You can see that we don't get the changes we expect when we do these changes, whereas the change occurs much more here. So Again, we don't know how general this is, and various toxins bind in various ways, but this is a demonstration of the fact that we're able to pinpoint the actual residue, the actual region that's in charge of binding to the membrane. And I don't have time to show this, but the Zarivach group went on to show this in other ways as well, using DSC and other thermal methods, where you can see melting of membranes that's affected by whether a protein is bound to it or not. And they were also able to show that mutating these positive residues and the interaction of PTA with the membrane changes. So we felt pretty confident that these results are correct, and that we do have properly pinpointed the membrane binding region. The second question I'd like to talk about a little bit more, because I find it really the more interesting of the two, and that is how the chaperone binds to the effector, and what does it actually do to it when it binds. Now, just to put this again a little bit in context, there's a thinking that the effector, in order to get through the needle, which is a relatively confined space, it cannot pass a structured effector. Remember, it's not just this domain, it's 650 residues. So clearly something has to happen to the effect in order to wiggle through that needle, if you want. And it's assumed and known, or it's pretty clear that there needs to be some sort of unfolding of the effector, if you want, as it goes through the needle. Bear this in mind. And here are two spectra. The first in black is a spectrum of our effector, the wild type regular situation. And the red spectrum is what we get when we add a 1-to-1 ratio of the chaperone. Now, two things are immediately seen. The spectrum becomes a lot less dispersed. It wasn't very dispersed to begin with. It's a helical protein, but it becomes even less dispersed. And also, we have several peaks that tend to disappear. This isn't surprising because the protein is getting a lot larger, but it was surprising that the residues that we expected to bind to the chaperone actually didn't show any sign of that. And I refer to the fact that actually the effector we're looking at, the 131 amino acid construct, is actually composed of a structure domain that starts at about residue 31 or 32 and ends at residue 120. And then we have a long N-terminal tail and a shorter C-terminal tail, which are completely disordered. This is by prediction. It's also by NMR data. Here is the relaxation data. Along the sequence, you can clearly see the difference between this domain here, which is structured, with low R1s, high R2s, high head NOEs, whereas these flanking regions look like they're flexible and completely disordered. Now, chaperones are thought to bind to effectors by what's called a globular/non-globular mechanism, which means that some parts of the protein of the effector, which are wrapping around the chaperone, are actually parts that are disordered. And the thinking was in our discussion between us and Raz and the group and the literature, was that we need to find this motif, which is sometimes called the beta motif, because it forms a trans beta sheet with the chaperone and the binds, should be looked for in this region here. And in fact, there was a sequence that seemed to be the right kind of sequence for this, but we could never find any indication of this. We couldn't find residual structure. And when we add the chaperone, we never saw a change in these residues. And that's highlighted here. Even times 50x, okay, these are the changes. The changes are minute. It's clear that the chaperone is not at all interacting with this region, and they don't disappear either. They just stay hanging there. So this was a surprise in the sense that where is the chaperone actually binding? It's clearly not binding where we expected it to bind. And that got us thinking, and that got a thinking, that we need to bring in reinforcements, because one of the great abilities, which I'm not sure I always have, but if I sometimes have, I'm very happy to be so, and that is know when your method is not going to be good enough for what you're trying to study, and you should be looking for a complementary method. And it was a very natural choice to speak to Sharon Wittstein, our EPR expert in the department, and Adi basically designed together with her this idea of let's label our protein with a few spin labels and see what EPR can tell us about this process of the effector interacting with the chaperone. Because if, as I should mention, that the chaperone itself is a dimer overall about 30 kilodaltons. So add that to the effector and add the fact that both of these proteins are hardly well behaved. Adi was pulling teeth to try to get these proteins to express and express in large amounts and label. So it seemed like we should go to a more sensitive method. And as I say, we turn to EPR. We labeled these 5 residues each once at a time in different proteins with spin labels in very standard methods, and we put them into the EPR spectrometer to see what we get. So here's our protein with the spins. As you can see, we've tried to spread them around the protein in such a way that they cover all sides of the protein if you want, so that whatever will be happening we'll be able to see. And the first measurements that Sharon and her group performed were CW measurements, where basically the spectrum is telling us something about the rigidity or the flexibility of the location of the spin. And you can see that in the 3 cases shown here, there was a clear change when we measure the spectrum with and without the chaperone. In the case of serine 45 and alanine 105, we have what looks like a rigidification, whereas with R95, we see what it looks like actually something that's becoming more flexible based on what the spectra look like. And of course, the natural extension of this is to think about doing Deer experiments, which measure the distance between 2 spins. So Adi designed 2 such pairs, one that contains the 77 and the 105 residues, and one that contains the 70 and 45 residues. And again, we measure the Deer experiment with and without the chaperone. And again, we got a big change, which was of course encouraging. You can see here the type of change we get for the first double mutant, but especially interesting was the change that we got for the second double mutant, the distance between S45 and A70. Because if you look at the data, it looks like there are 2 populations without the presence of the chaperone, but adding the chaperone basically collapses everything to the single population, and the one where the 2 spins are farther away from each other. And that's very, very convincing, very, very clear, which got us thinking that maybe there's more to this. Maybe actually there's a bit of a conformational change going on in the effector itself, and the chaperone is only picking out the conformation that it needs for binding. This is a time to go back to NMR, because as again, many of you know, NMR has developed methods, very, very accurate methods to follow these types of movements, of changes, of the shifts in proteins. And in this case, we performed relaxation dispersion measurements. Looking at how the spectrum looks, you can see it might be difficult to get good measurements for many residues. We use the methods suggested by Kenji Sugase, where you basically look at each amino acid NHP separately, and you focus on that only. In the conditions we were running it, each experiment like this would take perhaps, I would say, 6 or 8 hours to get a good spectrum. And we tried to do this on as many of the amide sites as we could. Bear in mind that I'm only looking at the structural domain of about 90 residues. And of those, I would estimate there are about 20 that can't be observed either for exchange reasons or for proline residues. And of course, there are others that overlap. But in the end, we were able to get to on the order of about 40 data points, which gave us good data that we felt could trust. And they're divided here into 3 categories. One category which shows really little or no RD effect. One which shows a large effect, but seems to be characterized by a lower K exchange value. You can see the relatively flat curve at the beginning and it only picks up later. And another category which looked like it had an exchange effect, but with a higher exchange rate, because you can see the effect really picks up earlier in the curve for those who are used to seeing curves like this. We looked at this a lot together with Miriam's help. And we're quite convinced that we have a statistically significant double process here, where it's wrong to attribute all these effects to a single change with a single K exchange, but we think it's more correct to look at this as something that's, if you want, biphasic with a double KX value. The KXs don't differ tremendously in rate, and obviously, if you know about central dispersion, you know they can't, because you wouldn't be able to see things an order of magnitude different, probably with the same method. There's a very narrow window which is appropriate for these measurements, maybe an order of magnitude and a half. In any event, it's about a fourfold ratio one will be about 1,500, one will be about 6,000 more or less. Okay, these are all fitted values based on many, many residues, and then phi x values are calculated based on these. But I don't want to be too technical. I just want to try to give some insight into what this might actually mean. So these are just two views of the same protein, just because I need to demonstrate different things. And shown in pink on the structure are those spin labeled residues, just like we get a feeling of where the distances we're measuring are by EPR. And if you look at the lower K exchange process, it focuses mostly on these residues. These are the ones that are most affected by that process. Again, the two sides are just two views of the same protein. Here are the residues that are affected by the higher K exchange, and you can see how they're really concentrated in one region. And here's what they look like together. So what struck us is that it really looks like all these changes are happening, if you want, on one side of the protein. It looks like the A prime helices are the ones that are becoming much more flexible, or they're the ones that are opening up, if you want. The change is there. The D helix, which is the tip helix, appears to also lose its rigidity and become flexible. Remember the R95 CW data. But it looks like after that, there's kind of a falling apart, if you want, of the shaft of the effector. And it really, in a way, exposes the hydrophobic core a bit, maybe not entirely. But it really looks like it's a significant change between these two conformations. And if you look at the right view, you can see how the protein is almost cut down the middle. Residues that are affected are all either on the right side or are touching residues on the right side. And that seems to be the governing process here. I'll try to speculate what this means. It's really very early at this point. We're working and publishing this now. This is all new work. But if I had to make a little jab, a little attempt to say what this means, it goes back to the globular/non-globular type of interaction that effectors have with chaperone. So here is a similar complex to the one we're discussing. This is a structure that was determined about a decade ago. It's the XSC-XSE complex again, chaperone, cognate pair of effector chaperone. And you can see how the effector doesn't really look like an effector anymore. It doesn't have a 4-helix bundle. It looks like it's really unraveled, and parts of those helices are now interacting with the chaperone. So you might postulate that something similar is happening here, where I'm only showing it schematically. We don't have the data to support this entirely. But instead of thinking of this region here, which we really prove is not the one that's binding to the chaperone, let's think of it in these terms, where here are helices, but at least one of them here, the A prime region, is opening up, and it's interacting with part of the chaperone, which would suggest what we call this globular, non-globular type interaction, where there is part that's flexible that's binding, and there's another part that stays globular. I would say very, very carefully, it looks like what the data we have is somehow a bit more consistent with this left possibility as opposed to this one, because we don't really see an unraveling in another region of the protein. It looks like we're looking at something like this. What this means as to, is this the beginning? Is this the very first initial event in the effector becoming unfolded so that it can go through the needle? Very possibly. If you think of it, it could really mean in a way that since the chaperone holds the effector in a slightly already unfolded state, it's easier for it to complete the unfolding process when it's time to actually go through the needle. But of course, we're nowhere near there. We're still focused on this very, very local aspect of the process of how the chaperone basically influences the effector, and how very possibly it's picking up on a conformation that's already there and not really inducing itself, but more stabilizing conformation that's already existent, even in our NMR tubing solution. So in the interest of time, I think I'll wrap up here. I've replaced the figure that we had here, which is a very general one, with what I would say is the main finding here of how the effector chaperone complex might look. And in doing so, I hope that I've been able to some extent, shed light on what happens in this interesting process of an interaction between a cognate effector chaperone pair. And I also hope that, by the by, while doing this, it was also a nice introduction for some and another demonstration for others, showing the power of NMR in addressing questions of this type. Again, I thank all those involved in the work. I thank you all for your attention. I'm happy to answer questions.
Okay. Thank you, Yarden, for this wonderful talk. And this talk is now open for discussion. And I don't know, there are some questions. And Thomas Schmidt, do you want to ask your question?
I see the question, so I can read it and relate to it if that helps.
Okay, this will be the best.
Yes,
Okay.
I have
Okay.
it. It's fine. So
No.
I'll ask for everyone to hear. Thomas asks, how many BTA are bound to the nanodisk? The reason I am asking is that the change in CW and gear traces could be explained by protein dimer trimer formation, even random interaction on the surface of the nanodisk as it is a defined space. I didn't make this clear enough. I guess I apologize. The all the CW and gear traces were all taken. There's no, that's not related to the membrane binding aspect of it. It's related to the binding to the chaperone. So there's some perhaps misunderstanding here, but it's not, there are no membranes or membrane mimics in those measurements. So that shouldn't be an issue. And we do know that the binding of the effector to the chaperone is a 1-to-1 if you treat the chaperone as a dimer. So there's no concern at all of having some sort of intermolecular interactions. We don't expect issues like that. And the second question also asked by Thomas, interesting to the binding of BTA to KP2 will be even more interesting to measure the R1 row and observe the dynamics of the protein upon binding to the nanodisk as described in a paper. Do you think the dynamics of possible subsequent protein interaction influence the activity of the protein? Excellent question. Actually, the spectra we see are definitely amenable to this type of approach because we're by no means seeing a very tight binding. It's a low affinity binding. So definitely the method you suggest could be used to track this down. That's something to definitely do in the future. I perfectly agree. And Supriya asks, what was the temperature for rotation dispersion measurements in slide 10? Did you try to measure the same at low temperature capture faster KX more accurately? Thank you for this question. We actually do have the data I showed was at 303K. We also have data at 283K, at 10 degrees Celsius. The reason we moved from 10 to 30 degrees was because we felt that we might be missing something that's going on by working at the low temperature for two reasons. First of all, it might be somewhat subduing or attenuating whatever dynamics are going on. And in addition, the spectrum was, for this type of measurement, was really very difficult to get good data. We felt that we were only able to look at maybe 10 or 11 or 12 residues and get meaningful relaxation dispersion curves for them. I will say that in general, the residues that we pick up at 10 degrees are quite similar to the ones, again, not surprisingly, the ones we capture at 30 degrees. And it's difficult to say if the KX values follow the relationship you'd expect, because I have to admit that these fits for the KX are kind of broad fits. And I think this is relatively common in the field. You don't really get an exact value. You can do all kinds of statistical manipulations to figure out what is your confidence region for this KX. But I wouldn't feel confident in saying anything less than maybe a twofold range in each direction. So it's difficult to say how the KX at different degrees relate to each other. The overall picture was the same. The temperature we chose seemed to be just give us more data. And I don't know if we could pick up these three different groups of behaviors if we wouldn't have data of that quality. So in that sense, it was the right decision.